Good evening, everyone. Welcome. Thank you so much for making your way out in this rainy night and getting your way through the puddles to come to the evenings at Whitney. First, I'd like to introduce myself. My name's Veronica Hinman, and I'm the incoming director for the Whitney Marine Labs. Thank you. And I'll be starting here January 1st, and I've been here for a couple of visits already, and I'm just here for another visit. I'm heading home tomorrow, actually. So I'm currently at Carnegie Mellon University, where I'm the Frederick Schwartz Distinguished Professor of Life Sciences in the Department of Biology and a professor in the Department of Computational Biology. And I've been the department head there for the last 5 years, and I'm now moving on from that role to be the director here at Whitney. Before going to Carnegie Mellon, where I started as an assistant professor, I did training at Caltech. And before then, I got my undergraduate degrees in zoology and, oddly enough, mechanical engineering from the University of Queensland in Australia, which is where I'm from, and my PhD in zoology from the University of Queensland in Australia. My research looks at cell and developmental biology, particularly from gene regulation and transcriptional control of cell specification using echinoderms, sea stars, sea urchins. So I've been using marine model organisms in my research for my entire career, and also studying regeneration, because of course echinoderms, sea stars and the like are very well known for their capacities of regeneration. So I'm looking forward to collaborating with people here and starting up my research program here with all the access to the wonderful resources in this local environment, and working with the faculty and getting to know the faculty. And so before I even leap into introducing Sandra tonight, I would like to remind you, just before you get to see today's talk, that October 10, we have another evening at Whitney with Sandra Exquisite is coming. She's a professor at Harvard University and also at the Howard Hughes Medical Institute. And she's going to give a talk about understanding multicellular life and the origins of that. And so that'll be a fantastic talk. And I know Sandra, I've seen her talk before. So hopefully you will come out and visit us again to see Professor Exquisite's talk. So, and my greatest pleasure now to introduce our speaker for today. So Sandra Loesgen, who is an associate professor here at Whitney Labs, and she's associate professor of chemistry at UF, and also of medicinal chemistry in the School of Pharmacy at UF. And she's been here since 2019, running her research lab here. Prior to coming to Whitney, she was at Oregon State University, where she was also an associate professor of chemistry. And she had a long, distinguished training career before that. She trained at NIH, had a stint at Howard Hughes Medical Institute at Genelia Farms. She was at the Scripps Institute of Oceanography at University of California, San Diego, and got her degrees at the George August University in Göttingen, Germany. If you look at Sandra's CV, she has a very long list of awards. She's been very busy and recognized during her research career. She has a Matt Suffinus Young Investigator Award. She's had a string of awards from the American Society of Pharmacognosy, okay. And at OSU, she had an award for outstanding and inspirational teacher in teaching in science. And you'll possibly see some of that come through in her talk this evening. And so Sandra does research on the diversity and bioactivity of chemicals produced by microorganisms in the terrestrial environment and in the marine environment. So she's interested in finding new drugs and compounds that will have medicinal applications by looking at these microbes that people aren't studying, and so a greatly unexplored area of diversity that has a lot of potential. And she's got this work funded by several grants from the National Science Foundation. So please welcome Sandra tonight, and I look forward to hearing her talk.
Thank you, Veronica. It's a great, great honor to be introduced by you in your visit here. So we are so looking forward when you start your directorship. And thank you all for being here. It's been raining out all day, so I'm very grateful to see a few faces, lots of them I've seen here before, Thanks for making the time coming out here to the Whitney Lab and bracing the rain. I want to take you today on a little journey, and I hope you have a little travel allowance with you. So I want to show you a little bit around the globe today, a little journey, and showing the microbes that we are excited for in my research group. And I try to convince you today that these microbes might be the source for the disease curing that we need in the future for many, many different diseases, from cancer and infectious diseases. I hope you can hear me okay. I hear a little double, but I think Sean is working hard. All right? All right, let's get started. Okay, so in today's seminar, I would like you to take around the globe, and that's actually really fun. We are following our microbes, the microorganisms that we are studying in my laboratory, and I want to take you on a little journey all around the place. I picked a few destinations, and I want to show you what microbes are interesting us. Why are they interesting for us to study? And what do they have to do with you and if you have an infection or know somebody who has a cancer disease? So why are these microbes important to our lifestyle? Before that, I want to introduce, it's a little bit of a lecture you might see, and hopefully you learn and take something home from tonight, is why does microbial chemistry matter? And a lot of people think, well, I go to the pharmacy and the pill I'm getting there, they come from a doctor. The doctor made that pill. Well, that's not always quite right. Many of these medicines that you are being prescribed are coming from nature. They're coming from plants, they're coming from microbes, and they have a function in nature. And I start often when I introduce this topic of pharmacognosy, which to all right is a very hard word, means we're finding pharmaceutical activity in the realm of nature. So what I want to show you today is a little bit of a history tour, and I'm a little of a history buff, so I hope I take you along the journey to introduce first these microbes and what they can do for you in terms of chemistry and disease treatment. And then we go on our little journey, stepping onto different continents and see what microbes have been found and what chemistry has been isolated and tested. So first off, a little bit of a lecture. I often show this slide deck here, and it's, I'm not sure if my laser pointer works. I thought I could deal with modern technology. Sorry, Sean, I pressed the right button, but I don't think it works. Anyway, you see two gentlemen there. They're a little older photo. Who in the audience has seen one of them before? Well, that's my lab, they're biased, they know too much. Anybody? Maybe the one gentleman to the right, he's actually quite famous. Anybody? Looks like a British gentleman. Oh, yes, indeed. On the right side, we have Alexander Fleming, and that might ring a bell for some of you. Alexander Fleming, with his serendipitous discovery of fungi that can prevent the bacteria he wants to study, actually, and he saw a zone of inhibition. So there are fungi around in the world that can stop microbes from growing, that can stop infection. And so that's what we see here in that slide. Alexander Fleming to the right, you see the fungus, the mold that gave us very precious medicine. Anybody knows what it is? Penicillin, wonderful, thank you. So penicillin, this wonder drug, came from this mold, and I have another slide to explain this, but from an organic chemistry perspective, it's a beautiful structure. I mean, look at that, it has several rings, it has heteroatoms. So as an organic chemist, I get excited. It's a small molecule, something that we can pop in a pill and take for a week when we have a bacterial infection. Okay, so penicillin changed the world, and I would argue it changed the amount of people sitting in the room here today. Why is that? Well, the discovery was made really, really early on, and that's obviously in all the textbooks, right? Alexander Fleming, looking at his bacterial assay, seeing a fungus growing there, that was a disturbance for his research. It wasn't something he wanted, but it seemed to have effects on his bacteria. So he described the effect of being antibiotic for the very first time, and that was in the 20s. So later on, it was tested, and that's where a researcher later on in Oxford, they injected penicillin into mice, living mice, and these mice had bacterial infections. And so they were able to save these mice from certain death. And that's also how all our drug discovery really started globally. People did not know there were germs. People didn't know that they need to use living organism before to take the first test patient. So really the establishment of using a mouse as an organism for testing, all established with penicillin. And it was mass produced really, really quickly in the 40s, and it has changed World War II, right? It was used. It can cure gonorrhea, 4 hours. Give it that time and you'll be cured for good. It was a miracle drug at the time. And yes, it was recycled from urine of soldiers. So all the soldiers in World War II at the Allied sites had penicillin pills in their backpack, and it saved people. And it came to this beautiful poster that you can find on many, many penicillin history books. Yes, your soldier, your son, your child will come home because penicillin will save it. So for us nowadays, that's normal, right? We can go in the pharmacy, we can get those tablets. But at the time, that was new, that was revolutionary. And like I said, it's estimated that 20% of the people here in the audience, in the whole population would not be here if it wouldn't be for penicillin. So it's a beautiful story, but I'm here telling you there are other drugs as well. And before penicillin came on the line, people were experimenting with dyes. And that's actually really fun because they figured out that this red dye that you see on top, that is called Prontosil. And that was actually the first antibiotic that was ever developed. So German synthesis chemists were putting together different dyes to making red, orange, yellow. And suddenly they figured out, if I give this to bacteria, these bacteria also die. They turn red before, but they do die. And that was one of the first tablets, as you can see here, in red powder. And later they commercialized them as the first drug that was available for bacterial infection. So penicillin started it all, and the sulfur drugs started it all. The idea we can find a cure, we can find a chemistry that can aid you in your health and in your wellness and can provide a cure to an infection or another disease. Prontosil was one of the first, as you can see here, the red color. It did color the patients red as well, but you know, I'm red, I'm orange, but I'm living. I do not have an infection that will kill me normally. If you're interested in this, I was very inspired when I made the slides by this book. Thomas Hager is a science journalist, a novelist, and he has this beautiful book that's called The Demon Under the Microscope. That is a wonderful read I can highly recommend. And it's just the ideas about the first antibiotic and the discovery that we made there. Okay, coming back to my introduction slide, the person to the right was Alexander Fleming, the person to the right, sorry, my left, your left. He is actually working on bacterial natural products. And so just to show you, Salman Waxman, he worked his whole career at Rutgers University here in the US, and he found the first treatments for tuberculosis. So streptomycin was one of the first treatments that were found by bacteria, soil bacteria. Here the macrolides, and here streptomycin. So for some chemistry connoisseurs among you, these are one of the most used antibiotics today in the clinic, all right? So there was no other treatment for tuberculosis, your lung disease that was so dominant in the 60s, 70s. These drugs made that obsolete. We can treat Mycobacterium tuberculosis with these drugs. So what's my point? Yes, we're going from bacteria and fungi to tablets that can help you cure an infection or a disease. And that's what drives our research group every day and drives many others to look for chemicals and compounds in the microbial world that we can use for human health benefits. And people are surprised when you show them these numbers. Many of the molecules that you find currently at the FDA-approved drug shelves, many of them are coming from nature. Who has taken aspirin? Yeah, that's on the top right? That's from plants. I hope nobody has taken Taxol. Taxol is from a plant, and it's a very potent chemotherapeutic. On the top right, we have streptomycin, that was a tuberculosis active reagent. And then on the bottom, we have the statins, the things that make us prone to get high cholesterol. The fungi molecules that we found were available to lower our cholesterol and allow the statins to be a treatment for high blood pressure and heart disease. So if you take one thing home today from this lecture, yes, the pills are in the pharmacy, but most of them are inspired by nature, either in form from plants or microbes. Many of the chemical structures there came from nature. One more shout out is to the 2015 Nobel Prize, which is a beautiful story. The microbe that makes these chemicals shown down here, ivermectin, this came from a golf course. So you don't have to go far. You don't have to go to the Amazon. This came from a Japanese golf course. The microbe was isolated. The chemical here is a beautiful structure. And this one is really the cure to many of the parasite diseases we have in Africa. So you see black flies can transmit little small microscopic worms, and they cause the river blindness, or they can cause elephantiasis. So diseases we see in Africa. You might say, well, I'm not traveling often to Africa, right? Why would I maybe care? This is the same drug that you might give to your best friend. Cats and dogs need these anti-parasite treatments, and these came from a Japanese golf course, from a microbe, right? Okay, so a little bit of a classroom style now. We think about antibiotics. We have them. We grew up with them. But that was not always the case. Like I mentioned, the first antibiotics were discovered as these orange dyes in the 30s. The sulfamides were the first there. Penicillin was next. I showed you from the fungus. And all the other names up here, all are from microbes. And you see the timeline. Once we realized there is chemicals that can cure diseases, we have to look for them, more and more was discovered. The structures of these compounds shown here, just to give you a little bit of, I saw chemistry tonight, and it cured people, right? These are beautiful structures. They're small molecules that these microbes are producing. Often we don't know why they're making them, not quite clear yet, but we can use them for our health and our benefits. Okay, so the timeline there is a little abrupt, and I can show you that in the future, we found more things. And many of the items on this timeline also come from nature, many, many of them. You also see that there's fewer discoveries in recent years, and you might heard about that. People talk about, where are our antibiotics? Do we find more? And the reason is every antibiotic will be, let me try this, but it doesn't, go. I can do by hand. Every antibiotic we will see and use in the clinic will give resistance at one point. And you see on the bottom now the years when on top we introduced the antibiotic to our health system. On the bottom, the year we saw the first resistant pathogens to this antibiotic. It's a natural occurrence. It's normal to happen. The microbe that produces many of these chemicals is naturally resistant to it, right? But it is something that shows us we always have to be on top of things, right? All these drugs, there might be a pathogen where this drug is not working anymore. So there is a problem with that, obviously, that we can't really keep up. And this was one of the first papers I found in 2004 that were talking about that. Like, how do we get more drugs? Then there are so many bad bugs. How do we do this? And anybody heard about superbugs, right? Lots of bugs that are resistant to maybe multiple drugs that we're using in the clinic. And maybe you have heard of a patient that enters a clinic and they test them and there is not a single antibiotic anymore that will work for them. And that's basically a hospice notice. These people might not leave that hospital because there is no drug for them. And why is that? Why do we not have more drugs, right? We should all have an interest in them. Why does our small research group here at the Whitney is pushing the envelope? Shouldn't that be Merck, Sandoz, GlaxoSmithKline, right? All the big names. Well, think about it. They are prescribing this drug for you if you have a strep throat and you take it a week. To make a drug takes billions of dollars, usually $8, $12 billion for one drug. And then one of you is taking it for a week. There's not much money to make, right? If one of you have heart disease, God prevent, you'll be on much longer medication. There's more money to make. And so from the big pharmaceutical companies, only very, very few are interested in giving you a new drug for a bacterial infection or fungal infection, right? Because if you get a fungus on your toenail, you might take it a few months. If you get a strep throat, you take it a week. But there's no really long-term investment. And the resistance is coming anyway, right? So very few companies are investing in finding new antibiotics. And the very few that are left are usually very small companies, many of them coming from academic university research. And obviously superbugs are expensive. Now, none of you, I hope, will ever experience one, but they are costing an economy a lot of money. There is loss in wages, loss in future health for sure. The World Health Organization was alarming for a long time about certain bacteria. You might have heard, you know, the dirty 12 escape pathogens. Those are like the super bugs that are in the news and media. And then to add on this is we only see very few new drugs. As you see, maybe remember the timeline, it got thinner and thinner in the end. Again, there's no money to make. And so many, many academic research labs are working towards a change. What you here see is a prognosis of the deaths that are expected in 2050 that are coming from antibiotic-resistant infection. And this will surpass the number of cancer cases, death by cancer in 2050, right? So we have a challenge here to find new antibiotics. And this was just a fact I found that how many are actually infected per year, and that over 35,000 are dying per year in the US by superbugs, by drug-resistant pathogens. So how does now the journey, you came here for a journey around the globe. You wanted to see nice pictures and exciting science. Getting there, getting there. But how do we connect that with soil? Well, most of the chemicals I showed you today come from fungi and bacteria. Things that live in soil, outside here, everywhere. And I love the statement from Da Vinci saying, and it's probably true, we know so much more about the stars, about our solar system, than we know about the soil underneath us. We don't know how many different microbes are there. We haven't digged deep enough at all. So that's what brings me to my journey with you today. We are interested in finding new chemical entities from microorganisms, and obviously we like to travel. Who doesn't like to travel, right? And so we are going and collaborate with people worldwide, trying to find bacteria and fungi that give us small molecules that can be drugs for the future. And I show you here the little selection. We have expanding our collaboration, and we're expanding our collection sites. So in the end of my seminar, I will bring you to Antarctica. That's the last place we've been sampling, very exciting space. But before that, I will take 2 or 3 vignettes, different locations, and show you the microbe, the research and why it's exciting, and what we think might be a drug lead for the future. I'll give you a little overview. That's the overview what we are doing in the laboratory. You might think, yeah, that's a microbe. Looks like a mold, right? Looks my old bread at home. How do I get from that into the pill that I want to pop and I want to, get in the pharmacy? Well, there's a process, and we have that here at Whitney established. We basically ferment our microorganism. Think about beer brewing, very similar. So we have our microorganisms, we grow them up. Smells like beer sometimes as well, not an unpleasant way. We look for what chemicals they're making, right? So we grow our microbes, we ferment them, we look at the chemicals inside, and now we need to decide which of the chemicals are useful, which are active, And that is, if I may, it's on the far right up, that the bioactivity, that's the area we would like to see early on. Sorry, I don't have a pointer. So on the far right, you see we have our microbes, we grow them, we extract them, and we want to test early on in the system, do we have an antibiotic? Is it worth our time? Do we have a cancer drug? Is it worth our time looking for? And then on the bottom, there's a chemistry part, which I don't bother you too much today, but that's where the fun part starts for us. It's a puzzle to figure out what molecules nature is presenting to us. So I have three short stories I want to share with you. The first stop is going to go back to the Pacific Northwest, all the way to the state of Oregon, where we had the pleasure to live for a few years. Lots of rain like here, which is not a good thing. Okay, well, we started off sampling soil in the Pacific Northwest. It's a very unique environment. It's a very dry area in the high desert, not in the coastline. And we went to areas that had volcanic activity, that had really high altitude soils that we could sample. And what you see here is the Lava Land State Park in Oregon, so it's really, really worth a trip, you should really go, where old lava tubes are still accessible. We can go into these lava tubes, we can sample unique soils there. We could also go to the high desert in Bend, Oregon. That's a very high altitude, high UV exposure area, and there we sampled the soils. And this is work done majorly with graduate students and undergraduate students at the time, and I have Zoe and Sandra shown on the slide. So we go all the way to Oregon, and there we try to stay away from the coast and the beautiful wine in the Willamette Valley. We went into the desert. We took samples from there, and we isolate the bacteria. And what do we find? Well, we found around 20 bacteria. Many of them help us in our antibiotic screen or in our cancer assay, but many of the compounds were known. And that's something I show here. Chemistry doesn't mean as much right now, but the color coding in green, all of these are antibiotics, and all in blue are cytotoxic, so they will kill cancer cells. So going in the soil in Oregon, we isolate bacteria that are highly potent in killing bacteria and killing human cells. Still blown away by this. Why is that? Why do these microbes make compounds that are affecting the other bacteria or humans? One of the molecules there was a little different, and I want to show you this molecule. This is on the far right. We named it bendy-terpenoic acid after the city of Bend, Oregon. And it's a beautiful little structure. It was one of the first bacterial terpenes that we found that have this bicyclic structure. And to our surprise, it was active against staphylococci. So these are gram-positive human pathogens that we try to inhibit in the laboratory. A little bit of a fun fact, when we look for the structure determination of these compounds, in solution, this molecule is actually quite bendy. Not just from Bend, Oregon, the whole molecule is bendy. And I'm not trying to give you a seizure here, but what you can maybe see is that on the far right, there is a ring system that we can actually wobble around. So this molecule has dynamics, has a little bit of a dance going on, and we can determine its structure in this three-dimensional space very precisely. And that's what you see here. It's been done by researchers in my laboratory. But even better nowadays, we can sequence the whole bacterial genome for a few $100 nowadays. And we can analyze the data. And with the colleagues at UF, in this case, Jeff Rudolph, we were able to find the gene cluster in the bacterium that makes this compound bendy-terpenoic acid. And we were able to put that in an E. coli host and make much more material than we would make otherwise from our natural product. And again, I mentioned it was the first of its kind, so this structure was very unique. And starting with this research, his team now found several other compounds that look like this 2 cycles that seem to be active in our Staphylococcus experiments. So that was my first trip. We kind of rushed over all the way to the Pacific Northwest. The next stop is going to Ohio. We're going to go and look into fungi that live with termites. That's an interesting story. Okay, this started a long time ago, actually, when Jason Slott, who's a professor at The Ohio State University, is asking us, I have this fungus, and that is not in the disease context. I have this fungus that I isolated in termite nests in Ohio. And that's what you see here. In those termite nests, and you see here these eggs in oval, there's a lot of fungal material in there. And I'm not sure if you can see this, but the oval things are the eggs of the termites. So these are wood-eating lower termites. And the little round balls are the fungal sclerotia. That's the material of the fungus. And this fungus lives there happily in the termite nest. The termites actually care for these balls. They lick their eggs, they lick the balls, they repile and reshuffle them from time to time. But the fungus is not taking over the nest. So the question was, why is this fungus living there? And it's called the cuckoo fungus because it puts its eggs, its little balls, into the nest of another organism, and the other organism is taking care of them. So we in the laboratory can take this fungus that you see on the far left there, and it's like a powder. We can harvest this powder off our agar plates, and we can analyze the chemicals in there. But the story gets a little more interesting. So Jason Slott is also really well known for the magic mushroom fungus. So he studies the genetics of psilocybin, and he told us, well, psilocybin, the compound in magic mushrooms, that seems to be genetically present in this fungus that you find in the termites. Can you look for me? We don't know what's going on per se, but there might be a party, right? There might be a mushroom party going on with those termites. We need to understand the chemicals. And it took us a while, but we were able to study psilocybin and now could exclude that. It's not really present in those termites, but what Annika Jagels, the postdoc at the time, found, I think is even much better. What you see here is our fungus in the middle. The powder, when you extract this, the chemical that you get out is the structure shown here. And that's something for us very exciting because it is a novel alkaloid. So it is a novel structure, nobody has seen it before, and the fungus makes it in very high yields, in very high amounts in this kind of powder. We also find the molecule being captured in a sugar molecule, so it has it with and without sugar. And that makes us interested in why is nature doing this? So we think about how this molecule might be released for its function in nature. Why are we excited for this molecule? And this is data we can now show after we patented with UF and had several talks with pharmaceutical companies. What you see here is a structure of the compound that we named after the termite fungus, Athelia pyrrolidine, that's the compound. And what you see on this really bar diagram over here is the binding affinity of this molecule to the serotonin receptor, the human serotonin receptor. And you might remember I mentioned psilocybin, so this is a molecule that stimulates the serotonin receptors. So we might not have found psilocybin, but we definitely found something new and unique. When we got the data, we got very excited. The first time we opened that email, we looked at that, we see a curve like that. And we're looking at it really careful and we see, wow, this other molecule, what they have as a control, that is a clinically used drug, an antipsychotic drug. It's used in autism currently. And our molecule from the termites in Ohio is almost as good, almost, not quite, but it's pretty good, pretty close in doing this work. It can bind to the serotonin receptor and has a similar effect than the clinically used drug. And then if you're a chemist, it has some benefits. We can make molecules. So we put the molecule together in our laboratory. It makes it also much easier to provide maybe grams and grams for future drug development. And so we put the molecule together. You see on top how we do it in a chemical sense. On the bottom, you see its signature in our spectroscopy. And yes, indeed, the natural product and the synthetic one are the same. So now we're in a business to start a drug company, I think. We did some more testing, and that's really important just to be sure. So on top, you see here how our molecule, Athelia pyrrolidine A, that was named after the fungus, how it fares compared to the clinically used drug clozapine. And we're very excited. We are in the 300 nanomolar range. So without any modification, this molecule came out of a fungus. We put it in human test systems, and it binds in nanomolar affinity. So that's very exciting and really rare, and we are looking forward to working more on this. We also figured out the mode of action. Does it bind to the receptor? Does it inhibit the receptor? Does it activate the receptor? And we were really happy to see, yes, it works as an antagonist, so very similar to clozapine, and it might be a drug in that direction. So you might ask in the end, what about the termites, right? There was a fungus, if I remember correctly, it was with termites, and it makes a serotonin binding molecule. Well, termites have serotonin receptors. We know that. But we try to treat the termites with our molecules, and that's what you see here. They're not really well collaborating with us, so we try really hard. Oopsie. Come back. Maybe no? Now you see it, very good. So we are trying to figure out, when we give this molecule from the fungus to the termites, how are they changing their behavior? Well, unfortunately, the termites are not really exploratory like ants are, or, you know, they show lots of behaviors like fruit flies. No, termites are rather boring. So we put it on filter paper that these termites hopefully take as food. And we put the compound on the filter paper, and then we recorded videos for hours and hours and hours, and that's what you see here. And we are still analyzing. Unfortunately, we are not quite sure what it does to the termites. I'm pretty sure the one to the far right, that's double the speed than normal, this termite on the far right is going for a marathon, right? I mean, there is something, and that's the one with our compound. So it should be a blinded assay, but we knew on the far right that is definitely our compound. And so we're analyzing this. We're not quite sure. There are not genetic tools on the termites right now to see what receptor they do or what the features are, but we're working on that. So we are working towards getting this drug. We have done it into mice, and we're talking with 3 different pharmaceutical companies. Serotonin targeting receptors is a big deal. It could potentially really come and develop into a drug, which is something that we all dream of in the basic sciences, right? A little bit more on the termites going crazy. Okay, I think that's one of the last stops before we go to Antarctica. And I want to take you home to my home country of Germany. I miss it sometimes, so let's go there. And this is a strain that is very close, a compound and strain that's very close to my heart. And I think some of my students always call it Sandra's favorite molecule. Because I am biased. I found that once. We really worked a long time on that. So let me introduce you to the strain and the molecule that we found, and we are very excited for. So what you see here in the middle is a structure of what's called Mensa carcine. And it's also from a soil bacterium. So one take-home message today is all the soils outside, all the fungus we might see after rain, they have cool chemistry. And many of the soil microbes make small molecules that could cure cancer or cure an infectious disease. So here in this case we have Streptomyces botopensis. It's a bacterium out of the soil, and it was found right next to the university where I did my studies. So University of Göttingen, right there in Germany, and every lunchtime we kind of walk out of the chemistry building towards the cafeteria, right? Good students go cafeteria. Cafeteria, for the German speakers, is Mensa, right? And when the microbe was isolated from soil right next to the cafeteria, the name was clear. It has to be mensacarcine. So it is named after the cafeteria. This microbe was found right next to the cafeteria at the campus at my old university. So when we worked with this strain, I was very lucky to recruit two fascinating and very good chemists. You see Elizabeth Carissa, she did a PhD, finished it here at the Whitney Lab a few years back. And before that, Birte Plitzko, she is a cancer biologist. And I am, the Mensa, so that's the cafeteria. And I'm very, very proud of this one paper that we got together, because it's truly a Herculean effort of these three women there trying to figure out what's going on. So long story short, this molecule is selective to kill melanoma cells, skin cancer. This molecule can kill skin cancer. And we did a lot of studies with this. And what you see here is a panel that the National Cancer Institutes are running quite frequently. It's called the 60 cell line panel. So every kind of cancer there is, every kind of tumor type has been tested in this panel. And what we found with mensacarcine is that it really hits the melanoma, the skin cancers. It doesn't affect the other cancers as much. This is pros and cons, but in latest research, we really try to hit a certain cancer specifically over all the other tissues in common. And in these curves, you see that for some of the melanoma cell lines that we have tested here, we really reach a lethal area. So we go all the way down at the lethal 50 concentration. Only 50% of the cells are surviving. That's where we want to be for curing melanoma cancer. Melanoma is a big topic in Florida, right after California. We are number 2 in the US in the statistics, right? So around here, protecting from the sun, very important. Following up with your dermatologist on skin is a very important thing. The numbers are actually not as big here. There are only a few thousand people dying of melanoma per se, but melanoma is one of the most metastasizing cancers there is. It might start somewhere on your skin, it might end up in your brain. So that's a very important thing to keep track of your skin and skin health. So the brain metastasis is a huge, huge challenge. And Florida, unfortunately, has the highest, second highest in the US. So what do we do in the laboratory? We do very simple cell-based assays. We ask cells, are you dying if we give you the chemicals? Are you dying a certain way? And that's what you see here in those gels. We are asking questions in the melanoma cells. Are you undergoing apoptosis with this controlled cell death? And that's something we want to see. We want to see controlled cell death in the melanoma cells, which are the first 2 bands, but not in any other cell line, like the colon cancer we have on top here. Again, it's a benefit to be a chemist. We were able to put a light bulb on top of our molecule. So what you see here is a chemical structure of our molecule, mensacarcine, and we added a fluorophore on top. And that makes it glowing red under the microscope. So we can follow this molecule, and what you see here is a melanoma cell. In blue, you see the nucleus, and we see our molecule, it's going into the mitochondria. These are this little green, yellow spaghetti. So we're going into mitochondria, we are causing cell death selectively in that cell type. That gives us a very exciting prognosis to explore. The next step we did is, and that came out of an interesting collaboration with Virginia Tech, with Amrinda 9 there. So what you see here is, these are human cells that are balancing on a very skinny tightrope. So there's a tightrope where these cells are kind of wobbling along. Cancer cells or many other cells are able to move and to have motions, and so that's what you see here. The cells are moving along. They actually try to grab those little wires and keep moving. And so we can study cellular motion with his assay, where we put cells onto this grid and see them moving, how fast they're moving, what force are they exerting onto that grid. Very cool science. What we see is, on top you see these little cells sliding along this grid. That's when there's no treatment, right? The cells can move along, they have normal motion. If we give them our compound mensacarcine, that's on the bottom video, you might not see much, but they're just vibrating. They're just not moving along. So mensacarcine takes away the motion of the cells. And I just told you guys that melanoma is the most metastasizing cancer there is. So taking away motion of cells is a very important thing to stop it forming new cancers. We did some other tests. Our mensacarcine has a similar effect if we take all the glucose away from cells. Glucose is what keeps our cells running, right? If we take the glucose away, we have the same effect as our chemical mensacarcine. So again, I want to give a shout out to Elizabeth. When she finished her PhD here at the Whitney Lab, and that was her last paper, when she looked into melanoma treatment very specifically. And the summary of her findings from this paper is skin cancer is a very, very serious disease. What you see here is a treatment of a skin lesions with a clinically used drug. The drug is called Zelboraf or Vemurafenib. It's two names depending on purchase or depending on brand name. And what you see here is that if you take this drug, after 15 weeks, you clear your skin cancer. You look almost cured. The problem is the skin cancer returns really, really quickly because the kinase, the BRAF kinase this molecule is targeting is mutating away. So you become chemo-resistant. This drug that worked so beautifully is not working anymore. And you see after 23 weeks, you're as bad off as before. So why is this important for our molecule? Well, our molecule still works when this cell lines that you see here on the bottom, and these cell lines are resistant to Zelboraf, to the clinically used drug. So mensacarcine, coming from this microbe in Germany next to the cafeteria, still kills melanoma cells when the clinically used, most often used drug, Zelboraf, is not working anymore. That was really exciting. We also see we can use these drugs together, and that's shown here. We can push this curve of death. We can push it to lower concentrations when we take our molecule mensacarcine with the current clinically used drug together. So more effect, more bang for your buck. So with the last two stories I want to finish, mensacarcine is truly my favorite, if there is a favorite among the chemicals. The latest news are quite exciting to share. This molecule is also very potent against parasites. So we just got feedback the last month about it active being against malaria, the malaria parasite. So cancer was one application. For malaria, we are actually much more effective. And as you might know, malaria is still a huge problem, majorly in Africa. If you ever had malaria treatment in the room, I hope not. It's a very unpleasant prophylactic treatment, so you will be very unpleasant side effects from all the malaria drugs there are. And the problem is many of the drugs don't work anymore. And so our molecule now, and especially during the pandemic, the numbers remain strong. So our molecule now, mensacarcine, and that's what we are talking now with people at Merck, is active in malaria, in the parasite. It's active against all the resistant malaria parasites that we have in our world, and it's currently developing drugs. So even drugs that we are currently developing that are forming resistance, our molecule is still working here. We have some other activity. Cryptosporidium is another parasite, so we are quite excited for that. Okay, for my last 2 or 3 minutes, I want to take you to Antarctica. I promised you a tour around the globe. I talk about chemistry, talk about microbes. I want to give a few slides to a place that I didn't go. I think it's too cold. But I want to take you along for the ride and show you that Federica went. So Federica, she just left. She was a postdoc in my lab. She took the opportunity in this January to go to Antarctica. And she took this beautiful photo showing the Whitney flag in the middle of the icebergs swimming around. So why did we go there? Well, we always have been, or had a long-standing collaboration with Jaime Cabrera Pardo in Chile. And I tried to cut the Google map so you see a little bit, a little tiny itsy bitsy tip of Florida up there. And then there's Chile. And then on the bottom here is Antarctica. You see it actually. And it's almost in the same time zone. Always pretty remarkable when you speak to people there. So he called me around December, on Christmas last year. He would like one of our team to go on the Antarctic expedition in January. Well, I didn't want to go. So very lucky that Federica took this job. So how does it work? How do you get to Antarctica? Well, again, we kind of go back. If you see Chile here, it's an extraordinary long country, right? It spans the whole continent of South America there. And so his university, Universidad del Biobío, is kind of in the center of Chile. But when you're gonna go to Antarctica, you go all the way down in the south. And that's what she did. So we had exactly 2 weeks to buy flights for Federica from Florida, going into Chile, and then from Chile going into Punta Arenas is shown here. That's the southernmost point of Chile, from where they start the expedition to Antarctica. We're working with the INAH Institute, the Institute of Antarctic Research of Chile, and they organized the whole trip and invited Federica and our team to come along. This is one of the last photos she sent out from Punta Arenas before they planned the trip to Antarctica. So how does it work? How does someone go from, a country like Chile then into Antarctica? Well, there's not really a normal flight schedule. So she arrived in Chile, she got trained, she got her equipment, everybody gets a red, warm protection suit. Everybody has the same, it's glowing brightly red. And then you wait for good weather. And if there's around 10 hours of good weather, there's an airline bringing you from Punta Arenas to Antarctica. And you might wait a week or two, because it's not always given that the weather is fine. And that's what you see here on the far right. When the weather is good, this one company, Chilean company, is flying from Punta Arenas a few times a day to Antarctica and bring people onto the research stations there. You don't need a ticket, because the company for flying there is owned or collaborating closely with the Institute for Antarctic Research. You see, everybody has the same red jackets, because they are provided by the Institute, so you look like little clones of each other. So how Antarctica? Well, here's a map. She's gonna go to the southern Shetland Islands. It's on the top right here. And we didn't know anything about Antarctica before she left. We kind of Googled a few things. Why is Chile actually so interested in Antarctica? Well, there is the Antarctican territories, right? Antarctica is its own continent, but it's not a country per se. It doesn't belong to anybody, really. So there are a few countries that claim territory, and that was put into a treaty in the 70s. And here you see those sectors. So the Antarctic, our southern continent, was just split by different countries. So Norway got a slice, United Kingdom took a piece, and then Chile, as you can see here, with the southern tip in Argentina, they're the closest really to Antarctica, and they also claimed a part of it. There's no really governing or governance there. It's really only like army and military installation from each of these countries. So if you happen to go to the Russian station and party, you gotta be persecuted under Russian law. If you go to the Chilean group partying, different rules, different laws, you have to be careful with that. I told her. So Federica went to the South Shetland Islands on this elongated tip there from Antarctica, and there into the Chilean. There are 4 permanently inhabited places in these islands that she went to Villa Las Estrellas, shown there. So starting off King George Island, she was then put onto a boat, or first from the plane, first on George Island into the plane. Here you see the main station from the Chilean collaborators in summer. kind of no snow. But that's actually a photo when she was there in January, which is supposed to be summer. So I don't know, still cold. From there on, they took a boat, and that boat is shown here on the bottom, to go to these different locations. And it's all for research purposes, for sampling microbes on different areas. I was surprised. She talked about a lot of animals. You could see a lot of plants, a lot of oysters. There is life in Antarctica. And in summer, for sure, you can see things. And one animal that she really fell in love with is this, what's called a stalked jellyfish. It's only present, there are 2 species that are present there, 2 of them only in Antarctic cold waters. And we took specimen with us here. We're actually looking into some microbiome studies. So it's a very beautiful, small animal that I have never seen before. I thought it was really cute. So we get a lot of microbes, a lot of samples coming from sediment, from ice, from algae, and from this little jellyfish. And then the work started. So once she came back here and the whole lab were helping processing all these different samples. And that's the outcome. From such a trip, over months and months later, we get these microorganisms that are filling our incubators. These are the ones we're studying right now in our assays for cancer, in our assays for new antibiotics. And they're just beautiful, so I hope I can show some around. So this really showed out to Federica, who was crazy enough within 2 weeks' notice to jump in a plane from Florida to Chile. I think it was a lifetime. She was in a helicopter. I think she wanted to go diving. It was just insane. So I'm so glad she went, because I would not go. And with that, I just want to, share our excitement and enthusiasm for the microbes. So I hope when you go outside, you can think about all the dirt. There's a cure, and there's a microbe. There's a lot of biodiversity we don't understand in soils, in waters, in the microbiome. And that's something that we try to preserve, we try to analyze, we try to study. And yeah, with that, I want to thank the team. This is one of the latest group photos. Very fortunate to be here at the Whitney Lab with all the colleagues. Great environment, and just wonderful to work with the students on these chemistries. And with that, thank you so much for being here at the first evenings.